And interneurons receive information from the transmit it to the other neurons. Again, it transmit to the other neurons. Motor neurons receive information from interneurons and transmit it to the muscles. And when we see the neuroendocrine cells function, the cell bodies of interneurons and motor neurons are aggregated with the fibers and interconnecting all types of nerve cells to form a nerve centers is called as ganglia. So here we see the main thing what we observe is the how the transmission of the pathway of nerve impulses and nerve fibers will be there from the axons to the dendrites. So here this figure shows you the how the cell body of interneuron, the pathway, how it is the axon and the where the this axon is going and joining the another neuron of the axon, and where the synapse is taking place, the region is called as a synapse, and that way the sensory neuron is connected to the cell body of the sensory neuron, or otherwise if the neuron is connected to the muscle, we will call it as a motor axon, where the motor neuron dendrite with the neuron, interneuron of the axon have an synapse region where the information is getting transmitted. So here we see in the insect nervous system also the central nervous system, visceral nervous system, and the peripheral nervous system. So coming to the brain part, we see how many ganglions are there in the abdominal region, how many ganglions are there in thoracic region, and brain, how many ganglions fuse and form a brain. So coming to the diagram explanation, the brain is a fusion of two ganglions which forms as an commissure. So it's at the subesophageal ganglion where this particular the nerves which are connected from the thoracic and ventral nerve cord will get united there to form an subesophageal ganglion connected to the brain. And in the thoracic ganglion, we see 3 ganglia. And in abdominal ganglion, we see 8 ganglions. So this part of the region where these 8 ganglions are, where they are located, it's a ventral nerve cord. And to them, they will find also the segmental nerves. So when we want to explain about the central nervous system, we say that it's a principal division of the nervous system and consists of series of ganglion joined by paired longitudinal nerve cords called connectives. When different ganglions are connected, we'll call it as a connective. If the similar ganglions are connected, we'll call it as a commissure. So primitively, there are a pair of ganglion per body segment. The ganglion of all head segments are policized to form a two ganglionic centers. So the brain and subesophageal ganglion. So the chain of thoracic and abdominal ganglion found on the floor of the body cavity is called as an ventral nerve cord. So coming to the brain part, we say that the brain or the dorsal ganglionic center of the head is composed of 3 pairs of fused ganglia. that is protocerebrum, deuterocerebrum, tritocerebrum. So when we talk about the brain, it's a dorsal ganglionic center of the head where composed of three pairs of fused ganglia. That is, the protocerebrum is associated with the eyes and thus bearing an optic lobes. So innovating the antenna, we found a deuterocerebrum. And concerned with the handling the signals that arrive from the body, we see tritocerebrum. And when we see the visceral nervous system, we'll find consists of three subsystems. That is stomodial, ventral visceral, caudal visceral, which include the frontal ganglia. And the ventral visceral and the caudal visceral, together the nerves and the ganglion of these subsystems innervate the anterior and the posterior gut, several endocrine organs, the reproductive organs, and the tracheal, including the spiracles. So this is about the visceral nervous network, how they'll have. So when we talk about the entire nervous system, it is enveloped in a connective tissue called the neural sheath or the neural lamella. And here we also see the fluid in the glial cells or the extracellular spaces. We find the higher concentrations of the sodium and potassium ions and the lower concentration of chloride ions and the hemolymph. So the nerves are bundles of axons invested in the neural lamella, and they are the glial cells that form a perineurium. So the nerves provide connection among the ganglia, between the ganglia, and other parts of the nervous system. We also see here in the neurons the examples of inhibitory cholinergic acetylcholine as a neurotransmitter, glutamic acid as neurotransmitters in the insect. So the insect nervous system is like this, where we are going to study about the peripheral nervous system. And it consists of the motor neuron axons, that radiate to the muscles from the ganglion of the CNS stomodial nervous system. The sensory neurons of the cuticular sensory structures, the sense organs that receive mechanical, chemical, thermal, visual stimuli from the insect's environment. So when we talk about the peripheral nervous system, all the nerves emerging from the ganglia of the central and visceral nervous system comprise of this peripheral nervous system. So the dendrites of these sensory neurons, where they are associated with the sensilla, these axons and synapse, where the synapse is forming with neurons. Within the ganglion of the central nervous system, and their nerves contain the motor fibers. And here we see the peripheral nervous system continuously inform the insect about its surroundings by receiving stimuli through the sensory organs. So these sensory structures are located all over the body, but are concentrated most on the antenna. So here, tarsi, palps, labellum, these are the ones. And the sense organs such as eyes and tympana also provide information. And the information about the external and internal environment is continuously carried from the sensilla to the central nervous system. And where it is integrated in a way that appropriate behavioral and regulatory changes can be made, and this interrelationship will be there with the various parts of the nervous system. So in the particularly when we want to describe about the nervous system of the insect, we first explain about the neuron. That is, neuron is the basic functional nervous system is the cell neuron. So, and this particular thing we will be explaining like neuron has a cell body and axon, and the neurons may be unipolar, bipolar, multipolar. That also we give the description, and we also give that what is the entire nervous system is enveloped in a connective tissue, and we also give the example of the neural lamella, perineurium, and the glial cells are involved in maintaining the composition of this particular fluid, what we were talking about, the sodium and potassium ions, how the level of higher concentration and lower concentrations are maintained. And the nerves are the bundles of axons invested in the neural lamella. And what are the inhibitory and excitatory transmitters we'll find cholinergic and glutaminergic. And we also give the example of the insect nervous system is divided into three major parts. That is the central nervous system, the visceral nervous system, and the peripheral nervous system. And in the central nervous system, we will explain about the brain, the double ventral nerve cords, and how the segmental ganglion join, and how the lateral and longitudinal connectives are there to that. And in the brain, we will explain about the three distinct lobes, dorsal, ventral, that is the protocerebrum, deuterocerebrum, and the tritocerebrum. In the protocerebrum, we will explain about the optic lobes, and the how the connectives of the optic lobes nerves will form chiasma, and ocellular centers where they receive the nerves from the ocelli and central body, and also about the protocerebral bridge, and also about the deuterocerebrum, which contains the antennal or the olfactory lobes. And here also we will be seeing the tracts of olfactory fibers connected to the antennal lobes and the corpora pedunculata. And we also explain about the tritocerebrum, which is the smallest part of the brain. And we also explain about the ventral nerve cord, giving the side heading as ventral nerve cord, where the ganglion adjoining the segments are joined by the paired connectives. Here we will explain about the thoracic ganglion, where two pairs of major nerves arise in the segment, where they are innervated into the wings. The wings are of mesothoracic and metathoracic. And abdominal ganglions, about 8 abdominal ganglions, and how they are getting innervated into. And also we will talk about the visceral nervous system. In the visceral nervous system, we will explain about the two endocrine glands which play an important role, corpora cardiaca and corpora illata. are connected with the nerves to the hypocerebral ganglion, and this ventral visceral nervous system is the one where we'll explain about the nerves which are innervated into the muscles and regulate the closing and opening of the segmental spiracles. And we also explain about the caudal visceral nervous system, where the caudal ganglion and the ventral ganglion supply the posterior portions of the hindgut region. And coming to the last, peripheral nervous system. Peripheral nervous system, we will explain about the sensilla and the axons, how they are connected to the synapse with the neurons, with the ganglion of the central nervous system. So coming to the physiology of the nervous system, we say that the conduction of the nervous impulses, action potentials, and often called the spikes, and the single sensillum to the central nervous system consists of an stimulus reception, transduction of stimulus to the receptor potential, and receptor potential produced via depolarization of the dendrite or the cell body. And like this, the action potential produced via depolarization is the axon of the sensory cell release the biochemical events at the postsynaptic membrane. So there ends the nervous system. Coming to the reproductive system, the reproductive system consists mainly purpose of the male reproductive system to produce, store, and to deliver the sperms, spermatozoa, and to produce the seminal fluid, which nourishes the provides an appropriate environment for the fluid and nourishes and provides the sperms to induce the female for the oviposition. And female reproductive system produces, stores eggs, provides eggs with the necessary nutrients for the embryonic memory development, receives and stores the sperms is the site of fertilization, and deposit the eggs and may provide additional protection to the usual embryos. So coming to the male, first one we'll talk about the female reproductive system. Basic what we will write in the reproductive system is the organs of the insects exhibit incredible variety of forms. There is a basic design and function, and reproductive system in insect can vary in shape, position, and in number. So between different insect groups and sometimes even between the different species into genus, genus, where the different species of genus will have an different groups of insects. So the female system, the main function of the female reproductive system are egg production, including the provision of an protective coating in many insects, the storage of the male spermatozoa, until the eggs are ready to be fertilized, transport of the spermatozoa to the female storage. So when we see the diagram, we'll say that it is an two ducts, how they are formed with the uh, ovarioles. We see the ovariole forming an germarium and the terminal filaments, and these paired ovaries will join and form lateral oviduct, and these lateral oviduct commonly join the common oviduct. And to that common oviduct, we see a gonopore and genital chamber and the vulva. And these particular things will have and spermatical gland joined to here, and even the accessory glands are joined to this. We also see the pedicel and calyx regions to the vitellarium, and in this way we say that the female system will have the main functions of the female reproductive system are egg production, including the pair of ovaries connected by the lateral oviducts to the common or the median oviduct. So the common oviduct opens posteriorly into the genital chamber, called as an vagina, after developed to form an bursa copulatrix, and it has an receives a penis during the copulation. And ovaries, when we see, they are bilaterally located mesodermal organs which produce the eggs, and the ovary is composed of number of functional units or ovarioles and covered by a lateral layer of epithelial cells along the muscles of large number of trachea. So the trachea provide necessary oxygen to the developing follicles for the maturation of eggs. So the number of ovarioles per ovary is normally constant for the species, but varies greatly in insects from one in the tsetse fly and some amphids, like over to 2000 in queens of certain termite species. So in most of the insects, each ovariole at a distal end is produced into long terminal thread that joins of its five neighbors, forming an terminal suspensory filament. And sometimes the filaments of two sides merge. So histology of the ovariole, we find apical germarium and basal vitellarium. And this particular one is covered by an outer ovariole sheath. So the germarium contains the oogonia that divide mitotically and become primary oocytes. And here we see the nutrients like yolk from the hemolymph and deposit in the mature egg. So the yolk taken up by the oocyte through the endocytosis. Types of ovarioles. There are two major types of ovarioles: panastic ovariole, meriostic ovariole. We come across them like this. See, this is an panastic, telotrophic, and polytrophic. These are the three types of ovarioles what we see, and we explain like this about them. So paniotic ovariole and the meiotic ovariole difference, where we see each developing oocyte is surrounded by an follicular epithelium and follicular plugs are between the adjacent oocytes. These are found in grasshoppers, crickets, and termites. Meiotic ovarioles has nerve cells. Depending upon the site of the nerve cells within the ovarioles may be either polytrophic or teletrophic. And after that, we will write about the growth of the oocytes. Oocytes growth takes place in two phases, slow phase or a rapid phase. What is the essential thing what we will find is the formation of RNA, DNA, protein, lipids, and some carbohydrates. So vitellogenesis is the last one what we will explain in the female reproduction. That is vitellogenesis in Drosophila. 1 lakh times during the vitellogenesis also may take place. So the vitellogenesis is where we'll talk about the yolk in oocytes, maybe a protein yolk, which is a protein carbohydrate complex, or lipid yolk and polysaccharides yolk. The protein yolk is more abundant and in forms of pitched deposit of protein in the oocyte. So the different types of yolk are derived from the different sources. So lots we describe about the pedogenesis. Pedogenesis also we have the slide. Coming to the male reproductive system. Okay, here still more is there, the explanation. Pedogenesis, where without the oogenesis, we will undergo the pupa or into the adult stages. We see most of the insects in and around. So this some special immature stages are capable of producing the mature oocytes, so known as pedogenesis, and ovulation will be there. So here in the ovulation, what we will write about the flies, how they function. So flies, the chorion serves as an protective coating, prevent water loss, and eggs when we want to see. When we see the egg shape and size, the mature insect eggs vary in different group of insects, but typically they are elongated and oval in longitudinal section. So oviducts, when we want to describe about the oviducts, the pedicel or the calyx opens to the lateral oviduct here, and the lateral oviducts join to form a common oviduct which serves as a communicating tube between the lateral oviducts and the vagina. The bursa vagina, which receives Eddygus. So here we also see the pouch-like expansion of the vagina, which receives the female ones as an Eddygus. The common oviduct and the bursa copulatrix or vagina are ectodermal in origin, therefore are lined with the modified tube. So when we see about the spermatheca, in most of the insects there is usually a single bag-like structure associated with the common oviduct. So in most of the insects there is usually a single bag-like structure associated with the common oviduct, where we see the sperms are stored for the prior to fertilization. So in flies, the number of spermaticae will be two or three. In grasshoppers, the spermaticae opens into the genital chamber independently of the oviduct. So accessory glands also we see the two pairs of accessory glands which usually open into the apical portion of the bursa copulatrix, vagina or common oviduct. These glands vary in structure and function. They usually secrete adhesive materials too. These glands vary structure and function, and the accessory glands usually secrete and adhesive materials to cement the eggs to the substratum or hold them together in ootheca. Coming to the male reproductive system, we find a pair of testes connected to the seminal vesicle with the vas deferens. So coming to the follicles in the spermatid, that is spermatozoa, vas deferens, how they will form with an apical sheath and apical cell called as a spermatogonia. And coming to the what is the difference between so the differences when we see the spermatogenesis and oogenesis? I'll show you the arrow diagram also, where the spermatogenesis when we take the mitosis it will undergo. So in mitosis, the primary mitosis, mitosis will form the primary spermocytes, and then the secondary spermocytes, and then the mitosis occurs and form the spermatids. So when we see the meiosis secondary spermatocyte, but is half the number of this thing, n. Mitosis spermatids also n. Spermatogenesis is also sometimes. So here in Oogonia also same primary, secondary, and vitellogenesis undergo the vitellogenesis. Here we see the all the nervous systems explained. Now coming to the oogenesis. So in insects, the internal fertilization takes place, which is generally brought about by the act of copulation. Most of the female insects are polyandrous and they mate several times in their life. And we see wasps, bees, flies, and bugs mate only once in life. They are called monandrous. Some species are polygynous. During the act of copulation, the semen produced in the male reproductive system is transferred to an appropriate site in the female reproductive system. So the seminal transfer may involve passage of either free semen or in many insects, one or more spermatophores also we see. And spermatophores are usually deposited by the male somewhere in the female reproductive system, that is bursa copulatrix or vagina, or rarely the spermatheca. So, however, aterigores, silverfishes, male deposits a spermatophore on the substrate and female picks it up and deposits it within herself. So, various mechanisms are involved for the release of sperms from the spermatheca. The sperms are either forced out by pressure applied by the female or by the mechanical perforation of the spermatophore. They are deposited, or in insects, the spermatophore may be digested away, causing the release of the sperms, or otherwise, sometimes in the form of free semen, the sperms ultimately move to the spermatheca. So, after the insemination, we see the fertilization. Fertilization process is involved with the three parts, that is, the release of sperms from the spermatheca, entry of the sperm into the egg, and formation and fusion of the male and female pronuclei. So, in monandrous insects, the sperms stored in the spermatheca will be staying for several months or years and are used to fertilize ovulating eggs in the.
Ma'am, PPT not available.
Yes. For this, the PPT will be like the diagrams have shown you, the explanation have been done. This is the only part where we are discussing about the insemination or the fertilization. So for this, you just go through the mechanisms for release of sperms from the spermatheca, how it happens in different insects, and that we will explain, and how the oviposition takes place. That thing we will explain in here. So I didn't prepare this particular two slides. So here in the most of the insects, the spermatical duct and its opening are surrounded by sphincter muscles. And these muscles are regulated by neurosecretions. That is, the neurosecretions are under the control of neurosecretory cells. And these neurosecretory cells are embedded in the muscles. So while we are talking about the monandrous insects, where the mechanism will be prevent from the sperm wastage, in Hymenoptera also, where the sons, that is, arhantocostly, we will call it. Arhantocostly reproduce they hive in the honeybees, that is Hymenoptera. Sons develop parthenogenetically by unfertilized eggs, while daughters develop by the fertilized diploid eggs. So unfertilized eggs, while daughters are developed by the fertilized diploid eggs in the Hymenoptera honeybee. So the mother is able to manipulate several factors like sex of each progeny during the oviposition by regulating the fertilization. So in this fertilization process, the factors which are more important are temperature, host complex, host size, paternal age, and the distribution, how the host is distributing the eggs. So influence the this particular regulation of the sperm release. And here the ovulation, the egg is oriented in the reproductive tract in such a way that the micropylar region in the where the close proximity to the site of the sperm release. So here, what happens in this wherever the micropylar region is there, sperm migrates to the egg, possibly responding to the chemotactically. One or more enter the egg and through the micropyle, wherever the sperm enters the egg, we'll call it as a micropyle. Here, more than one sperm also enters the egg, but only one fuses with the egg pronucleus, and the excess sperms usually degenerate, okay, without disrupting the development of the zygote. So we will say that the entry of the sperm into the egg, the egg nucleus undergoes the meiotic division, forming the female pronucleus. And here we'd also say the sperm that will fuse with the female pronucleus loses its tail region also, becoming the male pronucleus. The fusion of the toe pronuclei forms a zygote and signals the commencement of morphogenesis. So here we also say sometimes even the without fertilization also the insects may not be undergoing the same process in the same genus of same species of the insects, but the release of the semen, the copulation, the egg development maybe little bit vary in the different species. So most insects are oviparous. They lay the eggs on the other hand, and many insects are viviparous. They deposit different developmental stages. So the terms we will say that whenever they are having a different ways of laying and developmental stages also they are different. Larvae paris in the flies, nymphiparis in the amphids, and pupiparis in the few flies. So commonly used to refer the viviparous, larvae, nymph, and pupae respectively. So sometimes we also say that the term ovoviviparity is also sometimes used to refer the where the egg with the chorion develops, but the egg hatches within the parent before it is deposited also. We will see in some species of insects. So in favorable conditions only, the deposited eggs of the female insects, survival of the progeny takes place, and most of the phytophagous insects deposit their eggs on the surface of the leaves, often on the underside. Sometimes insects deposit their eggs under the surface of the leaves also, uh, to protect them from the extreme heats and natural enemies. So the eggs are cemented to the surface by an adhesive secretion so that they should not fall. Those are the accessory glands which will help in secretion. And we see some type of insects like locusts, the frothy material encases the egg mass, which is deposited in the ground, and where many flies oviposit in the surface of the freshly deposited cow dung by making a hole with its ovipositor. So, like that, also we see the oviposition, the laying of eggs in some of the insects. In the sawflies, these flies, sawflies, deposit their eggs in the inside the plant tissue also by using their ovipositor. So sometimes even the wasps, which are endoparasitic, uh, also they have an appropriate host, then only they will lay the eggs. Hoverflies, that is one which lays the eggs on the uh, endoparasite deposit, and we see the beetles laying the eggs inside the mounds of the dung, and we also see the aquatic insects like the mosquitoes laying on the raft like thing on the surface waters, or cockroaches deposit their eggs in the utica. Like this we find different way of laying of eggs in the different examples we can give for the oviposition. So they after the this thing we see the metamorphosis, how the growth will take place in the insect, and how this growth we will see a metamorphosis, where metamorphosis means will explain as an uh, how the developmental stages will be varying in different insects with the different types of stages. We will explain by types of metamorphosis, that is a metamorphosis, porometamorphosis, hemimetamorphosis, holometamorphosis, and hypermetamorphosis, where we will be the adult and the development of the egg laying to the adult, how the stages will be having a different uh, gonadial development, and how there is a variation in the external genitalia also we see in all of them, and a metamorphosis means example we see in the silverfish, where developing stages adults all will live in the same habitat, and only the insect will have a same structure from the um, larva stage to the adult stage. It possesses the same um, structure, only size will differ. And parametamorphosis we see in the leaf hoppers. The larva stage to the adult stage. The during the course of development, we see the wings developing like an wing pads, and the nymph stage, larva stage, and the adult stage. In the hemimetamorphosis, the insects like mayflies or dragonflies, stoneflies will show the hemimetamorphosis, where we see um, the immature larvae and the adults. There will be lot of specialized gills formed, or in if it is an aquatic insect, or sometimes if it is an fly, like we were discussing dragonfly. There is a quite um, the difference in the immature to adult when we see the hemimetamorphosis. Holometamorphosis means complete metamorphosis. Holometamorphosis. That is, we see in the housefly. Holometamorphosis. The stage of the larva, immature instar, is distinctly different from the adult, we say. And larva will don't have the eyes also, no mouth parts. They just survive, and after that they'll have the all the types of mouth parts and compound eyes develop in them. So this we will find even in the mosquitoes also. After the development only, there is no resemblance between the larvae and the adult. So this complete metamorphosis is called as an holometamorphosis. And the last one is hypermetamorphosis. Hypermetamorphosis we see in the examples of beetles. Beetles, where we see the here also most of the morphological legal formations will be totally different from the larva to adult. And this particular phenomena where we are seeing uh, much of the difference between the larva and the adult, where is no resemblance between them. This is called as an hypermetamorphosis. And like this, we come across 5 types of metamorphosis in the insects: ametamorphosis, silverfish, porometamorphosis in the leaf hoppers, and hemimetamorphosis in the dragonflies, holometamorphosis in the mosquitoes, and hypermetamorphosis in the beetles. Is it clear?
Yes, ma'am.
You have to join the another meeting, no? 10 minutes break I'll give.
Okay, ma'am. Thank you, ma'am. कृष्णवेणी लीव गाएगा. Hello. कृष्णवेणी.